Assalamualaikum and salam sejahtera. We are from group Leptospira Interorgans. Will discuss about experiment 7, diversity of bacteria. The objective of this experiment are to demonstrate gram-staining technique in classifying bacteria, to identify gram-positive and gram-negative bacteria, and to identify different shape of bacteria. The apparatus needed in this experiment are compound microscope, slide, wash bottle, Bunsen burner, bacteria loop, petri dish, forceps and staining rack. And the material needed are prepared slide of different type of bacteria, culture of Escherichia coli, culture of Staphylococcus aureus, yogurt, immersion oil, safranin, crystal violet, 95% ethanol, iodine, filter paper and labeling stickers. The procedure of this experiment are: we need to sterilize the apparatus before and after the experiment and make sure wear mask and glove. We must put a slide into petri dish. 95% alcohol will pour and soak for about 30 seconds. Then we need to use forceps to take out the slide. The slide was dry and heat by placing above the flame. Next, a loop of sterile DC water will place on the slide and a little bit of bacteria colony will put on the slide. Gently heat the slide to fix the bacteria onto the slide. Next, we need to place the slide on the staining rack. Remember, cover the smear with single drop of crystal violet and wait for 30 seconds to 1 minute. The slide will rinse with slow running water gently. After that, cover smear with 2 drops of iodine. Rotate and tilt the slide to allow the iodine to drain. Then cover again with iodine for 30 seconds to 1 minute. If we see the iodine does not mix well with water, this procedure ensures that the iodine will be in contact with the cell wall of the bacteria on the slide. Again, we need to rinse the slide with water as in step 6. Place several drops of 95% alcohol evenly over the smear. Rotate and tight the slide. Continue to add alcohol. Running from the slide appears clear to remove the excess stain. This is the most critical step of the procedure. If the smear are too thick or if the alcohol is kept on the slide for too long or too short a time, the result will not be accurate. Although there is no recommended time for this step, it usually takes 10 until 20 seconds to decolorize if exposed to a sufficient amount of decolorizer. Next, we need to add few drops of safranin on the bacterial smear and leave it for approximately 30 until 45 seconds. Colorless gram-negative cell will readily accept the light red safranin stain, while the already dark color gram-positive cell will undergo no change at all. After the step 9, we need to rinse off with water and blood dry with filter paper. To describe our observation in terms of type of bacteria, shape, color and determine whether it is gram-positive or gram-negative, we need to observe the slide under the oil immersion magnification. Next, repeat step 2 until 11 for microorganisms found in yogurt. After we are done with this experiment, we must wipe the table with alcohol. Do not wash your slide. Place it in the basin given. And remember, the whole procedure should be done in sterilized environment. And last, never rinse off anything regarding bacteria in the sink. Rinse in container given. So here the result that we get from the experiment using a microscope. That's all from us. Thank you.